and Phoebe will tell us about exploring the impact of environmental conditions on airborne Streptococcus pyogenes viability.
Great, thank you very much. I'm going to be talking about the impact of environmental conditions, namely relative humidity. I'll be focusing on today on airborne Streptococcus pyogenes. Streptococcus pyogenes, or group A Streptococcus, is an obligate human bacterial pathogen, which means it has to infect humans in order to survive. And you can see a culture of Streptococcus pyogenes on the screen now. So this bacteria can cause a wide range of disease manifestations, from more invasive diseases like necrotizing fasciitis to more kind of superficial infections like scarlet fever. And in the UK, scarlet fever is a notifiable disease and also a disease of concern. We've seen a significant upsurge in scarlet fever cases over the last 10 years in the affected population, which is young children. And these cases are mainly being attributed to a particular strain of Streptococcus pyogenes, known as M1UK. When we talk about M1UK, we classify it as an A3C pattern Streptococcus pyogenes, which means it's predominantly associated with throat infections. And this is a concern as because it's a throat infection, the bacteria is more likely to be emitted during respiratory events such as talking, coughing, shouting. So the predominant amount of research I've been focusing on has been on this M1UK strain. However, I've also been working on a strain known as M18, which is still this A3C pattern, so still associated with throat infections. But the key difference between the two strains is this M18 overproduces its hyaluronic acid capsule, which means it looks far bigger when grown in culture, and it's also far more mucoid. And we think about these when they're transmitted. There's kind of three key pathways we think of for throat infection transmissions with group A strep. The first one being direct contact. So an uninfected individual comes in direct contact with emitted bacteria. The second way is through indirect contact. So a person coughs, a droplet falls onto a fomite, and then infection is picked up through that fomite. But there's actually a growing body of evidence that aerosol transmission, so direct transmission of droplets produced during respiratory events, being inhaled by a secondary individual. But this is something we still don't really know a whole lot about for strep pyogenes. We don't really know what affects the bacteria while it's aerosolized. So that's something I'm hoping to investigate. And when we think about aerosol transmission, the key kind of factors that can impact it, relative humidity is really important. Temperature, we know, is also really important, and there's also growing evidence that gas composition can impact the pH of droplets produced during respiratory events, and therefore also impact pathogens present within these respiratory droplets. And as I said previously, my work currently is focused on relative humidity. And in order to investigate how relative humidity impacts strep pyogenes when aerosolized, I've been using a Celebs instrument, which allows us to study monodispersed populations of strep pyogenes droplets under controlled relative humidity conditions over time. And now the Celebs kind of operates in three kind of key steps. The first one being droplet generation from a droplet on-demand dispenser. So we load a known concentration of strep pyogenes into the dispenser and emit droplets from there. We then trap and levitate droplets for extended periods of time. This is where we kind of expose them to this set relative humidity we're interested in. And once our exposure is completed, we then deposit the droplets onto an agar slide, apply a small amount of liquid culture to disperse the droplets, and this allows us to see the number of colonies that have survived the levitation exposure. But in order to calculate kind of the relative viability, so the losses that we're seeing, we need to find an optimal baseline, our kind of 100% time point, to compare this to see the viability that was lost. So the first option we had was to take a baseline viability at a short time frame at a matching humidity. So this means the same humidity that our testing was going to be occurring at. And that kind of lets us see how viability varies over a set condition during a set condition over time. So we've done this with M1UK at 50% relative humidity. And what we see here is that viability remains relatively consistent, which suggests that any losses that we are seeing occurs before this first time point that we see here, which is 15 seconds. The second option that we had was to take a baseline viability at an assumed optimal humidity, different from the one that we were interested in investigating. In my case, that was 90% relative humidity. We see minimal losses due to evaporation at this time point. So we thought that would kind of give us the best bet of viability in an aerosol, but without any losses due to the condition itself. And what we see when we compare our time points to this is actually significant losses in M1 UK at 50% relative humidity over time, which kind of suggests that some of the viability may be linked with efflorescence of sodium chloride at this humidity. We know the media that we're levitating our group A strep in does contain sodium chloride, and at this 50% relative humidity, it's right on that efflorescence point. So the crystallization of the sodium chloride might be impacting cell wall viability, and that can cause death of the bacteria itself. But based on these kind of options that we found, we decided to progress forward and normalize to this kind of optimal condition to allow us to compare between different humidities that we're interested in. So when we do that with M1 UK, we see at 50% and also the 80% relative humidity that we have significant loss of viability over this kind of time frame we're looking at. But we see that at 30% viability, we actually have 30% humidity, that viability is actually maintained to a higher degree. And there's two kind of suggestions as to why this could be happening. We see in the literature that in desiccated conditions, so dried out conditions, Streptococcus pyogenes actually maintains its viability quite well compared to a liquid environment, so somewhere that has a higher water content. It suggested that in this kind of higher liquid content environment, group A strep maintains its metabolic activity and eventually begins to consume lactate and damages itself. Whereas in desiccated conditions, the strep actually lowers its metabolic activity and allows for maintenance of viability. The second option of to why this could be occurring is that aggregation in these droplets that are far smaller once they're evaporated might actually be able to maintain strep pyogenes viability, because the group A streps are aggregating together and kind of protecting the chains closer to the center of the droplet. So there's kind of two suggestions as to why we might be seeing this viability pattern. And when we compare this to that M18, that hyperencapsulated strain we're interested in, we see similar losses and similar changes in viability to what we saw in M1 UK, just to a lesser degree. And when we compare the two strains directly, we see at 30% and 50% relative humidity, there's not really any kind of significant difference between the M18 and the M1 UK viability over time. However, at 80% relative humidity, we do see a significant difference between the two strains, however, only at the 2-minute time point, which suggests this hyperencapsulation of the M18 might actually be providing some sort of survival benefit. We know during infection cycles that hyperencapsulation helps at M18s to evade neutrophil attacks from the human immune system, but that it might also be that this hyperencapsulation is actually helping maintain viability over time. So what are the kind of key conclusions we've taken from this? We see viability loss greatest at 50% relative humidity for group A strep, and some of that viability loss might be linked with that crystallization of sodium chloride. We also know that low humidities maintain Streptococcus pyogenes viability to a higher degree, possibly due to either decreased metabolism or aggregation of the bacteria. And we also see that hyperencapsulation could be providing some sort of survival benefit to M18s during extended levitation. And there's a large body of work we can do in order to kind of begin to understand why these patterns of losses occur. So yeah, thank you very much to my teams at Imperial and also the University of Bristol. Thank you.
Thank you very much. Phoebe, there is any questions for Phoebe?
Electrical charge of the droplets you continue to rise could affect variability.
Yeah, so there has been extensive investigation into the charging of the droplets in order to have them trapped within the EDT at the centre of cells. The charge that's being applied to the droplets is so low that it wouldn't really impact the viability at that point. So yeah, and there's also extensive literature investigating the charges applied to these droplets and also bacterial viability. It doesn't have any kind of significant effect.
Other teams did kind of thing work with over cell using acoustic waves to avoid electrostatic issue dealing with viability. So I don't know if you maybe have some opportunity to compare results between experiments realizing two kind of different cells to imitate particles.
Yeah, no, that would definitely be interesting to look into to compare with a different methodology.
Any other questions? Yeah.
Great talk. I was just curious as to what's happening with bacteria in those different conditions. Have you thought about doing any transcriptomics to see if there are some of the changes that you predict are happening?
Yeah, I think that's definitely something we are interested in looking into in the future. We don't really have any plans for that currently, but I think it'd be worthwhile looking into, as it's kind of an area of group A strep research that hasn't really been explored.
Hi, I have a question about when you're generating the aerosols, how do you know how many bacterial cells are within that aerosol so that when you're doing the CFU counts, that's accurate, basically?
Yeah, so when we are creating that kind of stock solution that we load into the dispenser, we use optical density at 600 nanometers. And we know from kind of the growth length of the strep pyogenes that it's in still an active log phase of growth. So we have a similar growth period. So we know they're not dead. So optical density can't really tell if cells are alive or dead, but we know based on the growth length that they are alive. And then we also have done some kind of outside of aerosol experiments to look at comparing optical density and CFU per mil over time. So we do have that data as well. But yeah, typically on a day-to-day basis, we just use optical density as it's just a quicker and a slightly more simple method to use.
Okay, thank you very much, Phoebe. It was a great talk.